All right, medical interventions for December 3rd and 4th. I'm going to talk to you about some of the stuff that you need to be working on outside of class. On December 3rd, which now I think all medical intervention students now are going to be on blue. It's going to work out that way. There are very few on gold, so they're all going to be on blue now. Um, so we will finish up that ELISA lab. That'll take at least probably half the period as we add the primary antibody, secondary antibody, um, in the substrate, and we see what those test results are. So that'll be the conclusion of that. We already, of course, we would have done this experiment prior, but I want to make sure you get that time in now. So I talked to you about 116. Here's what you probably see in Canvas at this point. Make sure you have the 116 outbreak analysis. So some of you have turned that in already, and that's awesome. So hopefully you get that turned in. Once again, that's due December 3rd, Thursday. All right. And then the 121 workbook, I asked you to get started on for the first 2 days here on the first blue gold days. And so you should have, by the time you come to class Thursday, you should have had this drawing done up here, right? Sorry, wrong. Yeah, this is it. This bacterial structure drawing. Oh, I took the wrong one. Let me open that other one up, please, real quick. 121 workbook. Yeah, so you should have had the drawing done of the bacterial cell. Okay, I'm going to talk about that a little bit. So this is going to be more of an informative type of video where I'm going to give you a little bit of background on some things. This video will help you as well. So I've made a little slideshow that's in Canvas. It doesn't really have any text in it. It's just a series of pictures. I'm going to talk through those as well. It will kind of help address some of these things. So in Canvas, under 121 workbook, you'll see 121 bacteria and antibiotics information. All right. So I'm going to go through this slideshow with you and talk about some of the things, the key factors that are in. So here's a tour of the bacterial cell. You see it is gram-positive. You have one bacterial cell will be either like this all the way or like this all the way. Nothing is half positive, half negative, right? This is what it looks like, okay? So you see the gram-positive side has a really thick cell envelope. Now, cell envelope consists of the cell, could be an inner membrane, but really the cell wall is the primary structure plus some other membranes that are in there. And it's primarily made of a chemical called peptidoglycan. All right, peptidoglycan. That's the thing that usually makes up that makes up the cell envelope. You see a really thick cell envelope on gram-positive, thin on gram-negative. Also, you'll see a couple other key points in here. This area here, which is really this, the arrow should be pointing to here for the nucleoid. That's essentially their DNA. That's the main DNA for the bacteria. Plasmids are little pieces, circular pieces of DNA we'll talk a lot about coming up. And plasmids are basically little pieces of DNA a bacteria can engulf through endocytosis. They can engulf and take in those little pieces of DNA. And sometimes they use it. So they eat another bacteria. It's a bacteria eat bacteria world out there. If they eat another one, they can take their DNA and incorporate it, not necessarily into their own nucleoid, but they can have this and they can use the genes that are on that. In biotechnology and chemists and biochemists have used these plasmids now to manufacture lots of drugs that are used for lots of medical interventions. You have the cytoplasm. You have ribosomes. Ribosomes build proteins. Right. The capsule is just basically an outer coating layer. It's like the first line of defense, but it's not very thick. It's not something that's going to create a lot of defense. You have these other structures called pili or fimbri, and these little structures that extend off allow for these bacteria to actually connect with each other and be able to actually move plasmids back and forth. So bacteria can connect with those. And then you also have this called a flagellum. So here's some of those structures that are in the diagram that you can see and what they do. So you can use a little bit of this to help identify what some of those things do. This gives you some more information on the plasmid. One of the things that is not in the diagram are endotoxins, and endotoxins are toxins that are secreted. Like we talked about meningitis bacteria, that they secrete toxins. And so those toxins are what often causes cell damage in the body when one of these things infects a human. So you can see those toxins are often located in just inside this layer and can be sent out. And typically, those toxins are more common in gram-positive bacteria. But we'll get into that a little bit more later. Bacteria also come in different shapes and sizes and different groupings. And so here you can see spheres are called cocci or a coccus. Rods are bacilli, or one is a bacillus, and then spirals are spirillum. All right, so you see spirilla, but the primary two are these. You have the circular shaped or spherical shaped one, which is a coccus. So you have like streptococcus and or bacillus, which are rod shaped. Okay, I don't need to know too much about that. I just wanted to give that as a background. And so then if you look, I'm going to move myself out of the way here for a second. When you do what's called a gram stain, so if you go back to our activity, so if you go back to our activity, and I'm going to pull this into another window, and you can see gram positive versus gram negative. So I'm going to talk through a few of these things real quick as we go. Hide myself completely out of the corner here. So gram positive is here, gram negative is here, okay? And gram positive, when you stain them, the gram stain technique, which is was found by a scientist named Hans Christian Gram in the late 1800s, started to look at staining mechanisms of how you could identify these bacteria, because you see these little rod-shaped structures, but the way, in this case, circular shaped structures, and whatever stain that they tend to take up gives you an indication about them. All right, so gram positive in the microscope, when you do a gram stain technique, they appear sort of purple. Gram negative tend to appear pink. All right, so essentially the staining process is adding stains on it. So you imagine you get a sample of bacteria and you put it on a slide, and then you add stains to that. After that whole sequence, gram positive end up purple because the stain is sticking to that peptidoglycan layer. And so if we look at the gram positive versus gram negative cell wall, start with a simplified diagram first. So here's that cell wall. Gram positive, or cell envelope is also what it's known as. Gram positive have this really thick layer of peptidoglycan. So you add the purple stain to that. All right. And then essentially you try to wash it away. Well, there's so much of this that purple stain stays and doesn't get washed away. This one, gram negative, that layer is so thin that the purple stain, yes, it gets absorbed, but it gets washed away. So in the end of the process, we had a pink stain, and so that shows up. The pink stain gets added to these too, but the purple is still there, so you don't see the pink. And so that's how you can tell. And so by seeing the color, ones that are purple, they have to have a thick layer. And ones that are thin, ones that are pink have a thin layer. The other indication here is that gram negative have two membranes. They have this outer membrane and an inner membrane. So this is the outside of the cell. They have an outer and an inner membrane. Gram positive only have an inner membrane. Okay? And so if you go back and look at that gram positive cell wall versus gram negative cell wall, more technical diagram, here you see a thick peptidoglycan layer with an inner membrane. And here you have an outer membrane and an inner membrane with the cell wall in between. Okay? So when you're looking at the document here, we talked about the colors. All right, some of these are filled in for you. Okay, and you can look at the drawings of some of those. You see some of those drawings on there. So, and then it talks about outer membranes, toxins. We'll talk about that real quick. Go here. So, gram-positive bacteria don't have that outer membrane, so they can absorb lots of materials like toxins. Versus gram-negative, they tend to not absorb those materials as easily, and so those thin layers of the membrane actually eject toxins. So toxins that go into the bloodstream to cause damage to cells. And so since there's this outer layer and this is really thin, things don't get into gram-negative bacteria as easily as they do gram-positive. While this is a nice thick wall, it's not very protective. And so things can get into the gram-positive and cause damage to them more easily than gram-negative. So these tend to be easier to kill. These tend to be harder to kill. So gram-negative bacteria infections are harder to treat. because of that. All right. So and there are some examples. If you look at some examples of different bacteria, actually, if you go back to the slide here, you can see some. I didn't put examples, gram-positive, gram-negative. Well, there's an example right here. So you have gram-positive Streptococcus, gram-negative E. coli. Do some research and you'll find some other examples. I don't want to give everything to you. All right, so do a few more examples. So like E. coli would be one of the examples here. And so a lot of the Streptococcus and Staphylococcus, they're going to be related to like upper respiratory infections and that kind of thing. All right. All right, so one more thing on this. I got interrupted there. Read through this for the class of antibiotics. You'll see mode of action examples for these different antibiotics. You have the beta-lactams, the sulfonamides, the tetracyclines, and the quinolones or fluoroquinolones are the ones that you're primarily responsible for. And fill that information out on the 121 workbook here to the best of your ability. And that is it. Okay, so you should be able to come up with all that. This asks you to kind of sketch that out. You may have to research how to sketch that out. So that would be pretty much it. So I'm going to kind of talk a little bit about that in class. Here's another slide too that will help you with it. So do the best you can on that. Let me know if you have any questions, and I will see you in class. Thank you.